Hi everyone. So it's been a little while since we've looked at some heavy bioinformatics. And the goal of last week's exercises and the goal of last week's review of protein structure was to solidify it in your mind, not just from a biology point of view, but from a bioinformatics point of view. Now, the science is the same. The basic biology does not change, but I'm hoping that some of the lessons we learned last time when we looked at the 4 levels of protein structure and then the PDB, I'm hoping that some of those exercises are going to be handy for you because today is a heavy day for bioinformatics. Okay? But protein structure analysis is also the most rewarding, in my opinion, topic that you could take up within the realm of bioinformatics. So that's what we're going to discuss today. We're looking at protein structure analysis algorithms, and most importantly, we're going to introduce a brand new concept called disorder and disorder prediction. Now, this is something that most of you may not have seen because it is not discussed generally in undergraduate classes. However, it is a very important concept, and it's a concept that's becoming more relevant to disease and understanding metabolic pathways and drug design. And so we're going to look very closely today at disorder. All right, let's get started. So as always, here's the overview for the plan for today. First, we're going to revisit the PDB and just quickly reintroduce that topic. It's not going to be anything new, but it'll segue us nicely into today. Then we're going to go straight into disorder before we look a little bit at crystallography. Then we look at other techniques of protein structure solution. And finally, we look at bioinformatics algorithms for structure prediction and for disorder prediction. Okay, so the PDB, just to recap very quickly, I'm not going to spend too long on this. It is the de facto repository for protein structure. These protein structures are solved using experimental techniques such as X-ray crystallography, nuclear magnetic resonance or NMR, electron microscopy, circular dichroism. There are many different techniques. Crystallography is the most popular one. And the structures you get are all deposited into the protein data bank. It started in the 70s. It was designed as a way to store this mass information before we had any concept of bioinformatics, when computers were in large rooms, or the size of large rooms, rather. There are 3 sites that you can visit it at. The RCSB, which is what you'll be looking at in lab. The PDBE, the European site, which is what I'm going to be using for a quick demo today. Just because it looks different, and I think you need to see what the different user interfaces look like, and the Japanese site as well. And this gave rise to the PDB file format, which is what we used to, at least you will be using some of it in lab today, to parse some data out using BioPython. And this is the format that was started in the 70s. And just a quick recap here, I had this question on the board last time, or on the screen last time. The PDB format will store sequence, structure, and other information. So here we're talking about the data, which is the sequence and the structure, and all the other stuff collectively is the metadata. So keep that in mind. And each PDB entry has a PDB ID that's 4 characters long. And this is what, when people are talking about structures, they will usually just refer to them by their PDB IDs. And I also showed you what an atom record looks like. This is a section of a PDB file which is describing the three-dimensional coordinates, so x, y, and z, of individual atoms. Now in this case, we have a set of atoms pointing to an aspartic acid residue, and here are the various coordinates for them. There's some other stuff down here as well on the right-hand side, but the PDB is a fixed width file format, so every single line, starting with this letter A, all the way down here to the other end, is a set size. It is the same length, okay? It does not exceed that length. And every section of the PDB has a standard for where exactly along the line certain information needs to be stored. And we're going to look at this a little bit later. All right, let's get into disorder. All right, so I know that last week's lecture was probably a bit of a recap for you in terms of biology, but you'll see why I did it that way. Because when we get to disorder, everything you've learned goes out of the window. Absolutely everything. And here's the question you want to ask yourself. Are proteins in nature, when they have structure, are they rigid? As in, are they like marbles? Or are they more like jelly? Or are they something in between? And the answer is, it really depends on the protein and what function it's trying to serve. And in fact, most proteins have regions, or domains, if you will, or motifs. I'm using all the terms that you should be used to by now. You have these regions that are not structured at all, in fact. So they're actually more like elastic bands, like rubber bands. And this lack of structure, and usually tertiary structure, is called disorder. So disorder is a region of no protein structure, and that's the key, okay? And this is a very large paradigm shift, because whenever you're referring to structural motifs, all of that discussion is completely useless when you're describing disorder. And in fact, how do you describe something that has no structure to it? So remember that this is a large paradigm shift, and it does make things difficult in bioinformatics and when you're solving structures, not just through computers, but physically when you're building crystals, when you're looking at NMR experiments. Disorder can really get in the way of that. And to complicate matters, we have proteins which have regions of disorder, which are partially disordered, and you also have fully disordered proteins. And these ones literally are like worms in water. So they're literally just moving around, adopting any kind of transient structure. They're like strings. It's like dropping a string in a bucket. So think of the bucket as a cell and drop a piece of string in it. That's your disordered protein. What is it going to look like? Well, it really is completely random. And these proteins, because they're flexible, they do not lend themselves to crystallization. So we cannot derive any semblance of structure for these proteins because, well, they don't have structure. But secondly, for crystallization, as we're going to see later, it's a very specific type of structure. And if a protein doesn't have any structure, it isn't going to crystallize. Partially disordered proteins, now you may get some crystallization, but as we're going to see later, the disordered regions you cannot view directly. So when you're looking at a 3D model of a protein, chances are you're not seeing the regions of disorder. Whereas in other cases, the regions of disorder may be expressed in a different way. Maybe you might see alternate arrangements. If there is a tail at the end of the protein, for instance, which is very common, you could see an NMR structure where you have a region of structure, so that's a globular protein as you're used to seeing, with one tail coming out at one angle and another tail coming out at the other angle. This is not to indicate that the protein has two tails, but that it's preferentially going to adopt one conformation or one shape or the other. But it's still very difficult for us to visualize this. So disorder cannot be viewed directly, at least through experimental methods. To further complicate issues, disorder can be spatial. So we have partial disordered proteins where a region of the protein is disordered, or fully disordered proteins where the whole thing is disordered. But we could also have temporal disorder, which means in certain circumstances, the disordered regions could adopt some structure. Think of a case where you have a boat that is about to be anchored, and you've got a chain with an anchor on it. So the chain with the anchor, if you throw it into the water, it's flopping around. That's disordered. But as soon as the anchor hits the bottom, hits the contact point at the bottom, suddenly the chain becomes very rigid and very tight. Now suddenly we know that this chain has order. It has structure. We can describe the structure of this chain. Similarly, as soon as the anchor is removed from the bottom of the water, bottom of the ocean or the lake or wherever you are, hopefully on a nice sunny day, now again the chain is flopping around in the water, and we cannot at any given time describe any kind of structure to this chain. It's the same thing with proteins. So if you have a disordered protein that binds to another protein, you could get some temporary order. But the point here is that we have to study it indirectly, and in fact computational methods are the only ways that are reliable for predicting disorder. And since we don't have any structural information, the only thing we can use for disorder is the sequence, and we're going to see that later today. But before we get into it, I'm going to quickly switch gears and talk a little bit about crystallography. Because X-ray crystallography is the most popular technique for solving protein structures and everything, and it's an incredibly heavy and dry topic. And don't worry, I'm not going to give you too much in-depth science into it. In fact, just as a side note, my doctoral thesis had some discussion on crystallography, and when my supervisor read my thesis, he said, "You now know more crystallography than your examiners do." So it is a science that is so heavy on reference material that there's no need to memorize the details of crystallography. But I'm going to give you a quick primer because you do need to know a few basic concepts. What is a crystal? Well, it is a protein or a single protein with multiple copies of the protein that are stacked together in a very particular pattern. So you can think of a crystal as an amplification of a single protein. But the thing is, crystals, protein crystals are not dry. They're not like diamonds. They're not hard. They're mostly composed of water or the aqueous medium, the liquid that the crystal was formed in. So they're actually more like jelly. They're more like a gelatinous, jelly. And in fact, if you can think of a cube of jelly, it's a bit wobbly. And that jelly, by comparison, has a lot more solid matter in it than crystals. Crystals are mostly water and mostly aqueous. So they're very, very fragile as well. But nevertheless, they are an amplification of a single protein. And so proteins in very special conditions, they form these crystals. And these crystals, again, are regular repeating patterns of these of the protein that hopefully the protein that you're trying to crystallize, that you're interested in, and not some other random protein that just happened to be in solution. Crystal packing is highly dependent on repeating structure. And because of this, impurities or extra, maybe a particle of dirt or some extra compound, maybe a little piece of carbon gets into your solution. The impurities are often eliminated because when you have a repeating pattern, there's no space for the impurity to sit in. Now, if the impurity does get into the crystal structure, it can make the crystal structure very fragile and the crystal could break apart. But if it doesn't, what you get is a crystal, a very pure sample. And so crystallography is actually, at its core, a purification technique. Keep this in mind, okay? crystallography is a purification technique. Then the crystals are taken to an X-ray source, and X-rays shine through these crystals, and it's like shining light through a diamond. And when you shine normal sunlight through a diamond, what do you get? You get a rainbow that comes out onto the other side that diffracts. X-rays do something similar, but you're not going to get a rainbow. You're going to get discrete dots onto the detector. And the pattern in which these dots diffract, or the X-rays diffract from the crystal, can be predicted through the principles of what's known as Bragg's law. Now, initially, I was going to put a diagram of Bragg's law, but really, I think that's going to be overkill. So just know that there's something called Bragg's law that determines how X-rays, and how any light, for that matter, diffracts through an object. Then these images that are taken of these dots are mathematically transformed. So these are called diffraction maps. They're mathematically transformed through a technique called Fourier transformation. And what that gives you is actually the individual coordinates, the three-dimensional coordinates of every atom in the crystal. Okay? So I'm obviously skipping many, many steps here, and the actual mathematics is incredibly complicated to the point that even I don't know it very well. But I certainly don't think, for the purposes of this course, you need to know much more than this. The point is, you have a crystal, an X-ray shines through it, you get a bunch of dots on a screen. It used to be photographic plates, but now we have high quality LCD detectors, and the images that you get are then manipulated through Fourier transforms to get individual three-dimensional atomic coordinates. And those are what you see in the atom records of the PDB file. Okay? But just note that X-rays will destroy the crystal very quickly. So most of these experiments are not done at room temperature, but they're done through a process called cryocooling, where the crystal sample has a stream of liquid nitrogen that's just cooling it down. And the X-rays will still destroy a crystal that's been cooled, but you certainly do extend the lifetime of the crystals. So large facilities will almost always use very, very cold or cryo-cooled crystals. And this is just a sample of some protein crystals. So they look like exactly what you'd think a crystal would look like. They have repeating geometric patterns. So just like you can describe the shape of a diamond, you can describe the shape of a protein crystal as well. And the PDB contains all this information. We're not going to get into it today, though. But I thought this was a lovely picture just to illustrate what individual protein crystals look like. So if you think about it, when just to go back to the purification point for a second, if this was all in solution and these proteins that are in these crystals were just kind of scattered around, when we induced crystallization, each protein of a certain type got together to form the crystal. So now you've purified this entire sample. So everything in this crystal, hopefully, is a single type of protein. Everything in this crystal is a single type of protein. Perhaps this crystal is trying to get the structure of a protein which is bound to some sort of metal. Perhaps this is hemoglobin, and you have a little bit of metal in there. But in any case, every single copy of the protein inside this crystal will be the same thing. That's in the best case. Creating crystals is not an exact science. We have very good guidelines, but even the best crystallographers will struggle sometimes because some proteins just don't want to crystallize. That's the best way I can put it. In fact, disorder really does mess things up here, because a region of a protein which is flexible could impair another copy of the protein to come close to it, to pack tightly. So disorder can actually disrupt crystal growth. But nevertheless, for the most part, you now understand the basics of crystallography. But here's a question: where do the X-rays come from? And for that, I get to show you a picture of a place where I used to work. And people in my department still collaborate with this place. This is the Diamond light source. It is very similar to CERN in Switzerland, which you might have heard of. And I believe there's another accelerator in Stanford that does the same thing. And this is what's known as a synchrotron. Okay, here's the word here, synchrotron. And it is basically a big donut. But what it's doing is very special. So at one end here, you have an electron gun. And this is a cathode ray tube, just like you had in your old, fat, flickery televisions. It's the same thing, but obviously on a much more powerful scale. Electrons get fired out through a linear accelerator, or LINAC. They get accelerated around this booster, and when they reach a certain speed, they get shot out into this bigger circle, the big donut over here. And that's called the storage ring. And there are magnets at different points here on the ring which bend the electrons and make them go in a circle. But when an electron bends, it gives off X-rays. So that's how you get the X-rays. And when the X-rays are formed, they get shot out into these tangential lines here. And each line is called a beam line because it's catching the electron or the X-ray beam. And each beam line can have different kinds of detectors to do different kinds of experiments. Not all synchrotron experiments are protein crystallography. No. They do a lot of cool stuff there. They study new materials for clothing. They look at space suits. They look at new materials for studying phenomena like the Higgs boson, subatomic particles. They do all kinds of experiments here at Diamond. Each beamline is designed for something different. And literally, a beamline is defined by the set of detectors or experiments that are inside the beamline. And I'm going to show you an animation of this. So this is from Diamond's webpage. So you have an electron that shoots in through the booster ring here, and then gets shot out into the storage ring. And each of these light blue dots here represents an X-ray going down a beamline. So that's basically how a synchrotron works. But the point is, when the X-ray comes out into the beamline, it is that X-ray that then gets tuned to a particular wavelength that the scientists determine are required for the protein crystals. And then the X-rays are allowed to interact with the crystal. So that's where the X-rays come from. And yes, if you think about it, to solve a protein crystal structure where the protein crystal is smaller than the naked eye, you need a facility that's this large to give you the power of the X-rays that you need to solve the structure. So it's pretty incredible and very poetic to think about it that way. By the way, Diamond is just south of Oxford. And we collaborate plenty with Diamond. I did most of my doctoral research down at Diamond, and I still do collaborate with them, potentially on a brand new project. And Diamond actually used some of the software that I wrote for my PhD thesis as part of their protein structure work. So very quickly, that was a primer on crystallography. I hope it wasn't too heavy. I tried to keep it as basic as possible. But I just want to point out that there are some other techniques available. Now, crystallography has obviously given us most of the crystal structures in the PDB. Almost all of them. But there are other techniques. So NMR is the next biggest technique. Electron microscopy, SAX, which is small angle X-ray scattering, circular dichroism. There are numerous such techniques, but the problem is that each of these techniques has some limitations. Hey, crystallography has limitations too. It's generally very hard to get a crystal structure, for instance, whereas NMR doesn't need any crystal structures, so proteins that don't crystallize may be better suited to an NMR experiment. But NMR has its drawbacks as well. But the point is, the PDB has structures that have been solved through all of these techniques. So here's a little exercise for you. After you do the PDB lab, perhaps, just find a PDB structure that's been solved by X-ray and one that's solved by NMR. And just go through the PDB file, go through the different pages, look at the kinds of different information you get between the two structures. It's not a homework exercise, it's more for you to understand the differences in the PDB files when you have protein structures that have been solved through different means. So how do we deal with disorder in the PDB? So I mentioned that the PDB has atom records, and the atom records contain three-dimensional coordinates for individual atoms. Now, if you're ahead of me here, you'll understand and you'll think immediately to yourself, there's something wrong with this. So let's take a look at that. The problem is that an atom is not going to strongly deflect or diffract X-rays if it's moving around. So if it's vibrating, or if it's part of a flexible region, a disordered region, and if it's moving around, the chance of an X-ray hitting it are lower than if it's in the same place. So you're not going to see strong diffraction or any diffraction from atoms that are disordered. So in most cases, there's not going to be an atom record for it. So if you look at the atom records, some atoms are just going to be missing. And that's just a fact. Now, in our case, we're looking at amino acids. And generally, the amino acids are small enough that all of the atoms in the amino acid will be missing. And in fact, in a PDB file, if somebody is able to get three-dimensional coordinates for a single atom in an amino acid, they'll probably just reject the entire amino acid and just say it's disordered. Okay? However, because partially disordered regions have proper atom records for all the bits that have structure, we can possibly obtain the list of sequences or the list of amino acids that are disordered. So think about that. We can use the structured residues in the PDB to determine which residues are not structured. Okay? And this is done through the combination of the secres sequence, which is derived from UniProt, and UniProt gives you basically the entire sequence that you expect to see in a crystal structure. And we're going to stick with crystals in this case. So all expected residues will be present in the secres sequence. But the atom record, since it's from the crystal structure, only the ordered residues will be present. So how do you get the disordered residues? Well, it's essentially secres minus atom, if you want to think of them that way. So let's use computing to isolate the disorder. Is there disorder in a crystal structure? So given a PDB file, so that's your input, you've got a PDB file, write some pseudocode, so it doesn't have to be Python or anything, for isolating all of the ordered and the disordered sequences. Now I'm going to do this exercise with you, but I think it'll be helpful to think about this problem in this way, and I think it's going to help you for the PDB lab that's coming up later today as well. We're not looking at disorder in that, but just to get your heads around thinking in terms of bioinformatics and programming, let's just do some pseudocode here. First, we parse the PDB file with BioPython. So now we know everything about the PDB file in memory. Excellent. Then we retrieve the secres sequence for each chain. Let's assume there's just one chain or one polypeptide to make this easy. So we have the secres sequence. Then we retrieve the atom records, and the atom records are per atom, but of course we can isolate a single atom from every amino acid, and we have the sequence of the residues in the atom records. And then we assemble the protein sequences for each chain. So in our case, we have just the one chain. And then maybe we just look at the residue numbers between the two and we compare which ones are missing. That's okay, that is not an exact solution, but I wanted to go through it to help you think like a bioinformatician. So now, as a second thought experiment, think about what other information the atom records have. And maybe you need to look this up, of course. And it may be easier when you do the PDB lab. But what other information do the atom records have to help solve this problem in a more exact way? So the solution I've provided here is not great. There are problems. I mean, there's no guarantee that the residue numbers are going to match. Remember, the PDB is from the 70s, and different experimenters use different ways of numbering things sometimes. It's very, very frustrating as a bioinformatician to work with old PDB files. But basically, when you're given a PDB file, you can't make any assumptions about the correctness of the file. So this is a good first approximation for finding these sequences. But continue thinking about this, and again, this is not something I expect you to answer today or tomorrow, or perhaps even by the end of the course. But hopefully, thinking in this manner, along with some of the exercises we do in labs, will help you come to some better solution for problems like this. But notice that this is a very, very simple, actually, use case for the PDB, BioPython, and studying disorder. So we've talked a lot about proteins that have no structure. So let's get back to proteins that do have structure. So what is structure prediction? Structure prediction is a methodology that there are different algorithms that do this in different ways, but basically you're given a sequence and it tries to predict, and the algorithm will try to predict regions that have perhaps secondary structure, so regions that are likely to be alpha helices. Tertiary structure is much more complicated. We'll come to that. Structure prediction is very useful when a particular structure is not in the PDB and you've got a sequence, you just kind of want to know what this protein could look like. There's a tool that you might come across very soon. It's called DSSP, Define Secondary Structure in Proteins. And it's not a predictor, but it looks at the sequence and it looks at the amino acids in the sequence, and based on the energy profiles of the amino acid, it assigns secondary structure, helices and sheets and things like that. But predictors will use this information to kind of start their prediction process, if you will. DSSP is some information that a predictor can use to help it make the best decision for determining how something is structured. There's another program called PDB-eFold, which is actually a predictor. It's a secondary structure predictor, which uses DSSP annotations, they're called, and it uses known similar sequences in the PDB to predict the secondary structure state when it's given a novel sequence. And in general, predictors will use some form of learning algorithm and some kind of a knowledge base, which they then use for novel input. So in PDB-eFold's case, it's using a learning algorithm that uses similar sequences and known DSSP annotations as its knowledge base. And then it applies it to novel input and gives you some kind of output. Now, tertiary structure prediction is difficult. Oh, I see a typo there. I'll fix that for you before it's uploaded, or for the final copy here. But tertiary structure prediction is difficult. It's extremely difficult. If you think about secondary structure being as hard as it is, tertiary structure is just almost impossible. But when computation gets better, algorithms get better. So there are some threading. So threading is a concept, is a type of prediction. It's a conceptual type of predictor that tries to simulate translation as the polypeptide comes out of a ribosome, and it predicts protein folding in that way. But it's not an exact science, and these algorithms have a long way to go. Okay, let's jump back to disorder prediction. So structure prediction is very hard, and disorder is kind of difficult to wrap your head around in terms of biology. But incidentally, disorder prediction is a bit easier computationally. Because when we look at an amino acid, we just want to know, is it ordered or is it disordered? We don't care about three-dimensional coordinates, none of that stuff. And so this is something called a binary classification, where a particular amino acid has two states. It can either be ordered or disordered. That's where the binary comes from. And there is a predictor called the RONN neural network. RONN stands for Regional Order Neural Network. That takes a protein sequence as input, and it gives you a disorder prediction for each residue. And why am I telling you about RONN? Well, this is the one that I developed, improved. So RONN itself was built in 2005. Well before I was working on it, but I built on it and I improved the prediction, and that was part of my doctoral research. And the new predictor is going to be released soon. But the original RONN is something that I still maintain as a web service, so we're going to take a look at it today. Nope. Before we get to the summary, let's take a look. So. Now, I built this web page. In 2005, they had a nicer looking web page. We're actually having some network issues, and we're going through a new server right now. So please excuse the fact that this page just doesn't look very pretty. I just wanted to get it working. But essentially, you have a sequence that is presented in FASTA header format. So there's your FASTA header on this line. And then you've got the sequence on the next line. And if you click the Send Sequence button, it takes a little while. So this is the old RONN, which is not as well optimized as the new one that I had a chance to work on. So instead of waiting for this to run, I'm just going to show you what the output looks like. Oops. Let's get over here. All right. What you get is a per residue disorder prediction that looks something like this. Okay? Each residue, so if you can think of this contiguous line as a series of dots, and in fact, let's zoom into a section here. Each of these is a residue, okay? And any residue which has a score above 0.5 is marked as being disordered. And anything below that is marked as being ordered. So we can quickly look and see that there are some regions of disorder here at the end terminus. There's a little piece here, there's a tiny little piece here, and a few more bits and pieces as we go along. And this is important when you're trying to crystallize a protein, because as I said, crystals can be severely hampered if there are large regions of disorder. Now, these regions don't look too bad, so a crystallographer might look at the RONN output here and say, I'm going to give this protein a try, because it doesn't look too bad. And it looks like these little bits here may not interfere too much. Other times you have large regions of disorder, and it may appear that you cannot crystallize the whole protein. So crystallographers will actually take parts of the protein. So maybe they only want to crystallize, maybe they only want to crystallize that region there between those two bits of disorder. And that's possible to do. So RONN gives you an easy way to kind of use this graphical output for perhaps planning your crystallization experiments. So that's kind of what disorder prediction is used for. And I know you have a lecture coming up on primer design where you're going to look and see how these sorts of web-based tools are used in the lab to plan out experiments. But this is a rough idea of how we could use disorder prediction. And in summary, computational structural prediction is extremely hard work, not just for the person writing the code, studying the problem, but also for the poor computer, because it has to do so many calculations and handle large data sets sometimes. Disorder is difficult conceptually, but once you get your head around it, is actually an easier problem as far as bioinformatics goes. It causes a ton of problems for crystallization, but handling disorder and predicting disorder is not too difficult of a problem. Now, disorder predictors and predictors in general for anything are not perfect. Keep that in mind, okay? Because they are learning-based approaches, they directly rely on a quality knowledge base in order to give better predictions. It's like me giving you a bunch of Spanish textbooks and then asking you to speak German. Well, that's not going to work out too well. But if I gave you a bunch of German textbooks and said speak German, now you're obviously going to do much better. Now, at the same time, if I give you a bunch of Spanish textbooks and a bunch of German textbooks and ask you to read some German, well, that's not so clear now, is it? Are you going to speak decent German or not? It kind of depends on the sentence, doesn't it? So predictors are not exact, but disorder is overall an easier problem to tackle. The PDB is the knowledge base for all structural studies. Keep that in mind. Okay? Most structures are derived through X-rays, X-ray crystallography, which is still the best method for determining structure, and it's getting better and better every day. Protein structure studies are very, very important for drug targeting, especially. So if you get a structure of a protein, you can look at the 3D structure and say, well, that portion looks like it could be good for binding. Maybe we can bind a drug to it. Then you go away and design a small molecule drug that binds to the protein and inhibits it. And this is obviously very important, especially now that we have Ebola running around and just absolutely massacring people. SARS, Ebola, all these large viruses and large epidemics and diseases, they relied heavily on protein structure studies in order to determine good drug targets. So that concludes my presentation. I hope that was helpful to you, and I hope you learned something new and exciting, and I hope it built up nicely on last week's lecture.